Hi, I'm Janine Cody, Director of the Chromosome 18 Clinical Research Center, and I wanted to go over the results from the survey we did of all the participating families regarding seizures. And everyone who's viewing this should have a paper copy of the report, so everyone who participated got a copy of that. And I think it's self-explanatory, but then I put it together, so of course it seems self-explanatory to me. So I thought I'd go over it really quickly, and hopefully that'll clarify any questions, but if you do have any questions, please feel free to contact us. So the survey was done last summer, but right before the conference, and we had a huge response. It was wonderful. You can see here that 192 people with 18Q minus participated, 91, whoops, I see a typo, 91 with 18P minus, that is, this should be 18P minus right here, 22 with ring 18, 61 with tetrasomy 18P, and 19 with trisomy 18. So the first page looks like this, and there were, and these are really just organized in an order in which they would fit on a page and be nice and neat. Okay, so there's no message in the order or anything that these are presented here. So first is trisomy 18. There were 61 responses. And I do need to say, I meant to say in the first slide, that if you're looking at the number of responses in each of these categories, the number I'm presenting in the summary for each condition may not add up to be the same as the number of the responses I showed on the first slide. And that's because we accept responses from anyone who's enrolled in the research. But when we're organizing the data in a way that describes, let's say, tetrasomy 18P in this case, we only use the data from those people that have the typical tetrasomy 18P with no extra 18Q material, not mosaic, that sort of thing. So we try to present the most unified picture. So those people's data are not included in this summary. However, those people's data is still really important to us. So it just depends on the question. So when the question is, what's the kind of classic picture, then we want the people with the well-defined molecular condition that's the same. When we have a question that's entirely different, that might be, how many people with tetrasomy 18P have this kind of seizure? Or everyone missing or duplicated for this gene, what kind of seizures do they have, or how many of them have seizures? Then in those kind of questions, then we would include everyone. So don't try to make the numbers add up precisely, but I want to make sure that everyone understands every single person's response is important to us, and it's never discarded. It's always important, and it helps in to answer different questions when asked in different ways. So, sorry, I meant to say that at the beginning. But anyway, so for tetrasomy 18P, we had 61 responses, and 33 of those had a seizure, had at least one seizure, so that's about half of them, and 8 had a seizure in the last year, which means 8 are having trouble getting them under control at this point in time. So for the first graph, regarding the number of seizures. So on the y-axis over here is on all the graphs are always the number of individuals. So the height of each bar is always indicating the number of individuals. And in this case, we're looking at number of seizures. So those with more than 10 seizures are in the largest group. So in tetrasomy 18P, people that have seizures tend to have quite a few seizures. That was 18 of them. 9 had between 2 and 10, and only 5 had only one seizure. Now moving on to the next graph, the age of first seizure. You can see the age of the first seizure for most people was between 1 and 5 years of age. Again, here we have the number of individuals over here for the height of each of these bars, and each of the parameters go across the bottom. So the first seizure can be at any age. You can see that can even be as an adult, but most of them occur in kids that are younger. The next graph is the length of the seizures, of the most severe seizure. And you can see that there's all sorts of different times from 30 seconds to less than 5 minutes to greater than 5 minutes. And most of them tend to have longer seizures, longer than 5 minutes. And here's the medication list. And I listed everything that everyone put on the survey. So the medications that worked are on the left here. And valproate worked in 7 people. But you can also see over here the medications that didn't work. And so valproate didn't work in 2 people. We, in our dreams, we were hoping that the medication list that worked would be one list, and the medication list that didn't work would be a completely different list, and we'd all know what medications to start with. But it just hasn't turned out that way. There tend to be some medications that work better, but they certainly don't work best in everyone, which really is what you would expect. It's not what we hoped, but that's reality. But this sort of list is something that can help your doctor help you figure out what's the most likely drug therapy that will work for your child. And the triggers, the most common trigger was illness or fever. But for a good number of them, there is no apparent trigger for the seizures. Moving on to 18Q+. So let me define this group for you. People with 18Q+, they don't have an extra chromosome, but they have an extra piece of the long arm, the Q arm, usually attached to chromosome 18, but in some manner they have an extra part of 18Q somewhere. Not a whole extra chromosome or part of an extra chromosome, or with a centromere. Their total number of chromosomes is normal. It's just they have an extra piece of 18Q. And that piece can be different sizes from different parts of 18Q. So this is a pretty heterogeneous group. And we had 4 responses, and 2 of those 4 had seizures. And both had more than 10 seizures, and both had a seizure in the last year. So in this group, those that had seizures had quite a few and had trouble getting them under control. And the duration of the longest seizure varied. One individual had a seizure lasting more than 5 minutes, so which means the other one had one less than that. And here's the list of medications that worked and medications that did not work. And so you can see that because they had trouble getting them under control, they have multiple therapies that they need to employ or try to employ to help get the seizures under control. So that's 18Q+. Now, going down the page to ring 18, there were 22 responses in this group. And 6 had at least 1 seizure, so that's about a quarter. And only 1 had a seizure in the last year, so that's good. That means most of them are getting them under control. And again, the triggers for the seizures were a fever or an illness. But again, quite a few had no apparent trigger. Going through the graphs again. Same order. Age at first seizure, again, 1 to 5 is the most common age, but you can see there were no adults who had a first seizure, so they tend to be younger, but most in the 1 to 5-year-old age range. Now moving on to the length of the most severe seizure. And you can see that they're less than 5 minutes, so they tend to be short. There are no greater than 5-minute ones here. And the number of seizures. So those who have seizures have quite a few, more than 10. So you can see that number. But again, the number of individuals is 4 here. Okay, so you always have to keep track of what numbers are over here on the left-hand side. So sometimes it's a small number. So although this bar looks huge, this is one individual had 1 seizure, one individual had between 2 and 10, and 4 individuals had over 10. So the numbers are pretty small. Oops, I forgot. The medications that work, again, are listed at the bottom. You can see the number of people for the medications that worked. So like Keppra had 2 and Topamax had 2. And the medications that didn't work are on the right. Moving on to trisomy 18. So this group of people is, again, not very uniform. The trisomy 18 group means they have 3 chromosome 18s, but some might have their third copy of chromosome 18 might have a deletion. It might only be the Q arm. It might only be P and part of Q. So they're not full trisomy 18 necessarily. Some of them might be. But as a rule, we enroll mostly long-term survivors, so they tend to not have typical trisomy 18. And also in this group might be some people who are mosaic for trisomy 18. So again, it's not a very heterogeneous group. And there were 19 responses, all over 6 years of age. So again, not a typical simple trisomy 18 group of people. 7 had seizures of the 19, so that's 37%, and 5 had a seizure in the last year. And the triggers were mostly no apparent trigger. Now looking at the age of the first seizure in years, again, they're all over the place from less than 1 to 11 to 21 years. The number of seizures, again, is most common to be more than 10. So those who have seizures have quite a few. The length of the most severe seizure, they tend to be short. So over 30 seconds and less than 5 minutes. So that's the good news. But again, these numbers are small with 3, 3, and 1 in these various groups. And again, the medications that work listed in the same way. The number of people on each medication that worked on the left, and the number of people for each of those medications that did not work on the right. 18P plus, again, like 18Q plus. These people do not have an extra chromosome, but they have extra 18P material somewhere in the genome. And so again, it's a pretty heterogeneous group, but usually it's the whole P arm that is most common in this group to have. And there were 3 responses, so small group. Didn't even make any graphs for the group of 3. One had 1 seizure lasting less than 5 minutes at about 2 years of age, and this individual is not currently on any seizure medication. So for 18P minus, there were 91 responses. 9 had at least 1 seizure, so that's only 10%. 4 had a seizure in the last year. And the triggers were, again, most often fever or illness, or no apparent trigger, which seems to be a theme here. The number of seizures are all over the place, but 4 of them had more than 10. 3 had 2 to 10. And you'll see throughout these, the numbers don't always add up because everyone didn't answer every question. So we used all the data we had, and so if people skip questions, then the number of responses aren't the same from question to question. The age at the first seizure, again, the largest number, which is 3, had their first seizure between 5 and 10 years of age. But again, you can see it can even be into adulthood. So first seizure could be at any time. The length of the most severe seizure, luckily, most of the time they're pretty short, around just over 30 seconds, but or less than 5 minutes. And the medications that work are listed under here. And you'll see the largest number of people are not on any medication at all, so 5 of those individuals. And at the bottom of that page, I wanted to be sure to mention, since we usually talk about 18p minus in 2 groups, those who have breakpoints at the centromere, so they're missing the entire p arm, and the other group who have breakpoints not at the centromere, meaning they have smaller deletions, some smaller part of 18q, always the end of the chromosome, but not the whole arm of the chromosome. And when we looked at the numbers and compared the 2 groups, there really was no difference. So in this case, we combined both groups because there were not differences. But what we also did was ask the question, okay, everybody with 18p minus who has a seizure, what is the region of 18p that all of those people have missing in common? And maybe that will help us pinpoint where a gene is that's responsible for the seizures. Well, here are those data in this picture right here. So we have a picture of chromosome 18 going across the top, and the red box, you've probably seen this kind of diagram many times before, or I hope you have. Red box is around 18p, just the p arm. So that means this box here is the p arm. And so here are the people with the smallest deletions who define this group. And so here's their study numbers over here. And here the pink bar shows the intact part of the chromosome they have. So here are people who have seizures, and here is their deletion. Goes from here to the end of the chromosome. Down here, these go from here, which is really the centromere, to the end of the chromosome. And this isn't everybody who had a seizure, because there were others who had other centromere deletions. But you can see the region they have in common is right here. This is the region that everybody with 18p minus who's had a seizure has missing. So we would hypothesize that one of these genes is responsible or plays a role in causing seizures. And here are all the genes. They're shown here, 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 here. Their names are listed next to them, or the acronyms for their names are listed next to them. They're shown going down the page here just so you can read them. If they were in a straight line in a row, then there wouldn't be any room to have their name next to them. So that's why they're scattered like this. But you can see it's quite a few genes, so this really didn't help us narrow it down very much. But it's still a critical region for a seizure, and it is helpful to us in our quest to figure out which genes cause which problems, so that we know can develop more focused and appropriate ways to treat them. Now, proximal 18q minus. Here I added a figure that's not in the handout just to clarify what proximal 18q minus is. So there are 2 groups of people with 18q deletions. Those with deletions here, and so here's the whole chromosome going across the top, and the red box shows the proximal 18q region. So all the people who have deletions in this region have interstitial deletions, obviously. Again, it's a heterogeneous group. Different people, no 2 people have exactly the same size deletion, but it's all interstitial deletions within this red box right here. And so we had 7 responses from people who had proximal 18q minus, and only 2 of those have had a seizure. And so you can just read this. 1 had a seizure at age 3. 1 had more than 10 seizures. And so we did the same thing we did on 18p. We asked the question, okay, of these 2 people who had a seizure with proximal 18q, what's the one region of the chromosome they both have missing? And that's shown in this red box right here. And the zoomed in view of what's in that red box is shown here. So here are the genes in that region, and with their acronym names listed next to them. Each little vertical bar, sometimes it's skinny, sometimes it's fat, is the actual position of the gene. So these are the genes in proximal 18q that 1 or more of these may be responsible for seizures. So just to clarify, as I move on to distal 18q minus, here's the same image from before showing the region of 18q for proximal 18q minus. There is a region on chromosome 18 right here. There is a gap between these two red boxes where no one has a deletion. So that's separated it into two very distinct groups, which is why we talk about distal and proximal 18q. So proximal 18q I just talked about. So next I want to talk about distal 18q minus. So these are people that have deletions anywhere of any size of this region of chromosome 18q. They could be interstitial deletions or they could be terminal deletions. And we've broken up the data into three separate groups because the gene TCF4, which is right here on 18q, has a very dramatic effect and we know is associated with seizures. We've separated out those data and into those people who have deletions that don't include TCF4 and those people who have deletions that do include TCF4. And so if a deletion includes TCF4, it could be interstitial, it could be right here, or it could be from here all the way to the end of the chromosome. So again, there's a variety of sizes of deletions, but we divided them into whether or not they include TCF4. And then we also have some data from people with deletions of only TCF4 and no other genes or mutations in TCF4. So now I'll move on to that. So going back to the handout then, for distal 18q minus, and these are people whose deletions include TCF4. There were 27 responses, 16 had had a seizure, so 60%, and 7 of those had a seizure in the last year, meaning they were not as well controlled. And the triggers, again, no apparent trigger, seems to be the most common answer. And the number of seizures, you'll see for most is more than 10, and that group included 9 individuals. The age at first seizure tended to be pretty young, either under 1 or 1 to 5 years of age. And again, here's the number. So this is 8 individuals had a seizure, their first seizure between 1 and 5 years old. 6 had one at under 1. Moving on to the length of the most severe seizure. And you can see the most severe one was less than 5 minutes. So most of the time they tend to be relatively short. But if your child is having a seizure, it seems like forever, even if it's less than 30 seconds. Medications that worked, again, presented exactly like the others. In this group, you can see there are several people who are on more than one medication in order to have their seizures under control. And again, you can see, for example, Tegretol worked for 2 people, and it didn't work for 2 people. So there is no common medication that works most often, but hopefully this list will help your neurologist help you figure out the best approach to take. And at the bottom of that page are people with TCF4 mutations, so or deletions that only include TCF4. So of that group, there are 11 responses, and 3 had seizures, so 27%, and only 1 had a seizure in the last year. And again, now these answers are all written out for you to read. And so again, medications that worked are listed here. No one listed medications that didn't work. So that would tell me that those who had a seizure got them under control with their first try, which is always a really good thing. Moving on to distal 18q minus, for people that had deletions that did not include TCF4. This group had 110 responses, so this is our largest group of study participants, and 40 had a seizure, so 36%. So if you recall, those who had a deletion that included TCF4, that group was 60%. If your deletion does not include TCF4, then the percent was 36%. So that's why we separated them, because there is such a big difference. 10 of these, so a quarter, had a seizure in the last year. As far as triggers, the vast majority had no apparent trigger. So I'll go through the graphs again. The number of seizures, there's nobody who reported only having one. So most of the individuals, now again, look at, here's the number of individuals. This is now 20 here. So 20 had more than 10 seizures. So those who had seizures had a fair number of them. The age at the first seizure, again, this is quite different from the other groups. This is 11 to 21 years of age. So in adolescence, most of them had their first seizure, and one even had their first seizure as an adult. So for the other conditions, they tended to be pretty young when they had their first seizure. And in this group, they tend to be older, which is an interesting observation. The length of the most severe seizure, again, is less than 5 minutes or less than 30 seconds. So most of the time, they're relatively short. And again, because this is a group of 110 responses, the list is very long of medications that worked and medications that didn't work. But the interesting thing is that 11 of these individuals have never been on any seizure meds at all. So, if you have any questions, we're happy to answer all the questions that we can and tell you when we can't have an answer. Sometimes your questions are the ones that make us think about something differently or give us an idea of the questions we should ask in the future. So please ask your questions. The person to contact is Anna Hill, who's a project manager, and she will funnel all those questions probably to Dr. Atkinson, who's our neurologist. I'm definitely not a neurologist. And she will bug him until he answers and see that you get your answer. We appreciate your support with this is all about collecting information from families and getting it back to you so that you can make the best decisions possible for your children. Because, of course, our goal is to make the chromosome 18 abnormalities the first treatable chromosome abnormalities. And we're well on the road to that, thanks to the fact that we're all working together. We're all in this together. Thank you so much. Have a great day.